1859, had shown in his natural selection theory that all organisms reproduce many more offspring than survive. Thus, he estimated that a single pair of elephants would have over 19 million offspring alive after 750 years. Cormondy, 1978, mentioned that a single pair of English sparrows after being introduced into USA could give rise to 275, 716, 983, 698 descendants in 10 years. Howard estimated that the female Musca domestica on the average produces 120 eggs at a time and there are 7 generations per year. Assuming that all survive 1 year and all the females 50 sex ratio produce each generation, then at the end of the 7th generation, there would be more than 6 trillion flies. Dash and Carr, 1990, found that Lepidopilus olivacea, the olive ridley sea turtle, female lays, on the average 110 eggs in the Orissa coast. Assuming a 50 sex ratio, a female after 1 egg laying session is replaced by 55 newborn females. A female lays eggs many times in her lifetime. If all the offspring survive, 1 female can be replaced by a few hundred females. All these examples indicate the potential of organisms to reproduce. This reproductive capacity of the organism is called reproductive or biotic potential. The term biotic potential was coined by Chapman, 1928, to designate maximum reproductive power. He defined it as the inherent property of an organism to reproduce to survive, i.e., to increase in numbers. It is a sort of algebraic sum of the number of young produced at each reproduction, the number of reproduction in a given period of time, the sex ratio and their general ability to survive under given physical conditions. Thus, with the term of biotic potential, one is able to put together natality, mortality and age distribution. Each population has the inherent power to grow when the environmental conditions are not limiting with regard to space and food, and interspecific competition is absent. The growth rate per individual in the population may reach a maximum and remain constant under specific environmental conditions. In a population with a stable age distribution, the reproductive capacity or growth rate is called the intrinsic rate of natural increase, represented by the symbol R. The is the exponent in the differential equation for population growth in an unlimited environment under specific physical conditions. In fact, the index R is the difference between the instantaneous specific natality rate, i.e., rate per time per individual, and the instantaneous specific death rate and may be expressed as R equals BD. Life table for the 1952. Thus, intrinsic rate of natural increase. Organism approximate biotic. An equilibrium will be reached either by decreasing the birth rate, natality, or by under natural conditions. Biotic potential is a rare phenomenon, since environmental conditions do not permit unlimited growth of any population. The difference between the maximum R biotic potential and the rate of increase which occurs in an actual laboratory or field conditions is often taken as a measure of the environmental resistance, which is the sum total of environmental limiting factors which prevent the biotic potential from being realized. Environmental resistance is often low when a species is first introduced into a new territory, so that the species increases in number at a fantastic rate, as when the rabbit was introduced into Australia and English sparrow and Japanese beetle were brought into the United States. But as a species increases in number, the environmental resistance to it also increases, in the form of organisms that prey upon it, parasitize it, and the competition between the members of the species for food and living space, and consequently, population decreases. An equilibrium will be reached either by decreasing the birth rate, natality, or by increasing the mortality rate. Popuation dynamics. Populations have characteristic pattern of increase which are called population growth forms. Such growth forms represent the interaction of biotic potential and environmental resistance. The study of population dynamics is done by 3 approaches: 1. Mathematical models, 2. Laboratory studies, and 3. Field studies. The growth is the most fundamental dynamic feature that a species population displays. Populations characteristically increase in size in a sigmoid, S-shaped or logistic fashion. When a few organisms are introduced into an unoccupied area, the growth of the population is at first slow, positive acceleration phase, then becomes very rapid, logarithmic phase, and finally slows down as the environmental resistance increases, the negative acceleration phase, until an equilibrium level is reached around which the population size fluctuates more or less irregularly according to the constancy or variability of given environment. The level beyond which no major increase can occur represents the saturation level or carrying capacity. The carrying capacity or equilibrium density is represented by the letter K. It is often useful to define the maximum rate of growth of the population. This parameter, generally termed the intrinsic rate of natural increase, is symbolized R0 and represents the growth rate of a population that is infinitely small. Accordingly, such type of population growth can be described by following logistic equation. dN dt equals R, NK where R equals innate capacity of population to increase birth rate without resource limitation, N equals population size, and K equals highest population density that can be maintained in real environment, i.e., at carrying capacity. There are 2 main types of population growth forms. 1. J-shaped and 2. Shaped or sigmoid forms, figure 6.8. I-ill. The growth forms are due to the nature of I. Species and prevailing environmental IF. Limit. Conditions. In J-shaped curve there is a rapid increase in density with the passage of time, called exponential growth. The density J-shaped curve. Density values when plotted against time. Give a J-shaped growth curve and at the peak the population growth ceases abruptly. Due to environmental resistance. 4. Example, the population growth curve in human populations and growth of yeast. Drosophila and rabbit under laboratory conditions show an initial slow rate and then it accelerates and finally slows, giving the growth curve which is sigmoid or S-shape. The peak constant level represented by K or upper level, called asymptote, of the sigmoid curve is called the maximum carrying capacity. It marks the limit to which the environment can support the population. Time. Figure 6.8. J-shaped and S-shaped population growth curves. Plant population dynamics. In many respects, plant populations behave like the animal population, but they have some unique features such as follows. Most higher plants are modular organisms, developing from a single zygote but producing an indeterminate number of repetitive structures, called modules vegetatively. A clump of herbs, grasses or trees may be product of one zygote. Plants cannot move to mate or disperse. Thus, they have evolved means as gravity, wind, water flow or animals for dispersal of pollen, seed, vegetative parts, etc. The seed population present in the soil for different species are referred to as seed bank or seed pool, Silvertown, 1987. All these seeds do not germinate or all the seedlings do not establish. Some die due to environmental stresses and this is called environmental sieve, which allows only the stronger individuals to survive. In most cases, the seeds germinate in batches and seedlings of one lot is known as cohort. In this way, from a huge seed bank through ecological selection, cohorts are formed and these in turn result into adult population. This process is referred to as recruitment. Further, a plant may originate from a vegetative part, called ramette or tiller, or from seed called genet. Thus, ramette and genet form two levels of population structure. The term clone is normally used to designate the population derived from ramette of the same parent plant. The three-halves thinning law. Most aspects of growth of population are density related. One important generalization applied is three-halves thinning law. If we plot the relationship between the dry weight and density of shoots, known number of individuals of in plant population, the line relating weight of each individual to density has a slope of 1.5 or 3/2. The slope would be 1 if increasing density has been exactly compensated by reduction in weight of individuals. Thinning is normally inversely density dependent, but does not always occur if the growth of the plants is extremely plastic. The three-halves is universal and applied well in a wide variety of plants from mosses to trees. Exact reason of its occurrence is still not clear. Growth rate of population. The rate of growth of a population is expressed as the number of individuals by which the population increases divided by the amount of time that elapses while this population increase is taking place. Number of births B, number of deaths D, average population in time interval growth rate R equals the actual change in population number AN over any span of time at is equal to Rn. A is the entity that is changing. This can be written AN AT equals Rnr. Using the symbology of the calculus, the rate of change of the population at any instant time, dn dt, can be expressed dn dt equals Rn. This is equivalent to saying that the number of individuals at any arbitrary time t, or nt, is related to the number of individuals at the beginning, n, by the equation nt equals ne, where e equals 2.71828. The base of the natural logarithms. If risk constant, the growth of population will be exponential. If risk positive b greater than d, the population shows an exponential increase to indefinite density, and if r is negative b less than d, it shows an exponential decay to extinction. It is impossible for a population to change at an exponential rate indefinitely. However, there are many cases in which conditions are such that b is substantially larger than d for a period of time, following which conditions change so that d becomes much larger than b. The responses of populations to variations of this sort is an exponential population explosion during favorable conditions, followed by a crash when conditions change. Diatom populations in Lake Michigan, USA, for example, undergo such exponential increases at different times of years, triggered by variations in abiotic factors within the lake, followed by equally rapid declines. Popuation dispersion. Populations have a tendency to disperse, or spread out in all directions, until some barrier is reached. Accordingly, population dispersion is the movement of individuals into or out of the population area. It takes three forms: emigration, one-way outward movement; immigration, one-way inward movement; and migration, periodic departure and return. Dispersion supplements natality and mortality in shaping population, growth form and density, and also it plays a significant role in the distribution of plants and animals even to the areas previously unoccupied by the members of the population. Most types of population dispersion occur due to a number of reasons such as for obtaining food, avoiding predators, preventing overcrowding, result of action of wind and water, environmental factors as light and temperature, breeding behavior, physiological reasons as secretion of some hormone or for interchange of genetic material between populations. Emigration. Emigration under natural conditions occurs when there is overcrowding in the migratory locust, lemming, grouse, snowy owl, snowshoe rabbit, Arctic fox, gray squirrel and occasionally in other species. This is generally regarded as an adaptive behavior that regulates the population on a particular site and prevents overexploitation of the habitat. Further, it leads to occupation of new areas elsewhere. By dispersing into new localities, there is opportunity gained for interbreeding with other populations leading to more genetic heterozygosity and adaptability. Itis, of course, population pressure that is responsible in large part for the dispersal of the young and extension of ranges into new areas. Under normal conditions, adult animals, especially among the higher vertebrates, are well established on their territories and the youngs are forced to seek homes elsewhere. Among insects, there is a relation between emigration and inherited behavior tendencies. Individual tent caterpillars, both larvae and adults, differ innately in the extent to which they show activity even within same colony. In the development of populations of excessive size, spread of infestations of the insect into new regions is large by the more active individuals. The outbreak finally terminates when the proportion of sluggish individuals comes to predominate in the population. Wellington, 1966. Continuous emigrations are rare and when they occur, result in depopulation. Equilibrium of populations is maintained in such circumstances by enhancing the reproductive ability as well as by decreased mortality among the populations. Immigration. Immigration leads to a rise in population level, causing an overpopulation which may lead to an increase beyond the carrying capacity. These immigrations result in increased mortality among the immigrants or decreased reproductive capacity of the individuals. Both emigration and immigration are initiated by weather and other abiotic and biotic environmental factors. Migration. Migration is a peculiar kind of population dispersion which involves the mass movement of entire population. This can occur only in mobile organisms and best developed in insects such as desert locust, Schistocerca gregaria, migratory locust, Locusta migratoria, butterfly, Danaus plexippus, and in the migratory dragonflies, Libellula quadrimaculata and Pantala flavescens, in fishes like eels, in birds and in certain mammals. Most two-way migratory movements are arthothermic processes of population. Migration of population may occur for food, shelter, or reproduction. And regular periodicity is a common feature. Very often, environmental periodicities control these migratory movements, as for example day and night rhythm, lunar periods, tides and changing seasons. The monarch butterflies, Danaus plexippus, travel very long distances and their migrations are found to be pathed every year through their same conventional routes, and their migratory movements are initiated by the oncoming winter and the return trip being influenced by spring. In most cases, migration of population may occur for food, shelter, or reproduction. Better utilization of uninhabited or hitherto untouched habitats and their resources are the greatest benefits derived from the migratory movements. However, during migration of population, mortality of numerous individuals may occur due to different ecological hazards such as temperature fluctuations, scarcity of food, predation, etc. Anyhow, migration has certain benefits for populations, as it enables wider dispersion of populations, it avoids intraspecific competition for food, shelter or any other means. Regulation of population size. The inherent tendency of all animal populations is to increase in number. But this increase in number is, however, not infinite since the carrying capacity of the environment always imposes a restriction upon it. Thus, after reaching the carrying capacity level, the population density tends to fluctuate above and below this level, and such fluctuation in population between upper and lower limits tend to give some stability to the population. The nature of the processes that regulate the numbers of plants and animals is a major problem of population ecology. Many groups of animals and plants are provided with unique and intrinsic self-regulatory mechanisms such as failure of reproduction and self-inflicted mortality for controlling the size of the population. Populations grow when natality exceeds mortality, and they decline when mortality exceeds natality. However, limitation of animal number in a population is brought about by the action and interaction of two basic regulatory processes, namely density-independent and density-dependent factors. Density-independent factors are the extrinsic factors which tend to regulate the density of a population under different conditions, appearing to act on the population and inflict loss of individuals irrespective of the population density. Variations in space or cover, favorable weather and food occur independently of population densities and may cause drastic changes in the abundance of animals. Such ecological or environmental factors influence negatively or positively all the individuals of a population irrespective of density. The density-dependent factors are intrinsic or biotic factors, and they depend on coaction between individuals within the same population or between populations of different species. Density-dependent factors may stabilize populations at an asymptote, the level of which is determined by the carrying capacity of the environment. Some of the important density-dependent factors are competition, reproductivity, predation, emigration, and disease. The combination of factors or any specific factor involved in the density-dependent action may vary from species to species. Further, these species have been known to display the property of intercompensation. According to intercompensation, if there is a change in the environment to relieve the population from the pressure of an existing effect, then the population increases till it reaches a level when the second effect takes over. Thus, if the predators that normally keep herbivorous animals down are removed, the population of herbivores may increase to become overcrowded and result as starvation. A supply of abundant food at this junction would make the individuals susceptible to diseases due to the intensity of crowding. Wilson and Bossert, 1971. Generally, the population fluctuations controlled by extrinsic factors tend to be irregular and correlated with the variation in one or more major physical limiting factors such as temperature, food, water, etc., while fluctuations of populations controlled by intrinsic factors exhibit regularity and population cycles. Population cycles. Populations are said to be cyclic when they alternatively erupt and subside in a more or less uniform manner between high and low levels of density. Different animals exhibit population cycles different times. The best established cycles of population density of fluctuations are those of periodicities of 3 to 4 years and 9 to 10 years. The 3 to 4 year cycles are most commonly observed in many birds such as snowy owl, willow ptarmigan, capercaillie, black game, hazel grouse, etc., mammals such as lemmings, voles, arctic foxes, etc., and fishes. 9 to 10 year cycles of population density fluctuations have been 9 to 10 year cycles of population density fluctuations have been observed in birds such as ruffed grouse, sharp-tailed grouse, willow ptarmigan, etc., and mammals such as snowshoe rabbit, muskrats, Canada lynx, etc. Among invertebrates, insect pests of coniferous forests in Germany fluctuate in periods variously from 6 through 18 years. Asterias forcaceae is found to contain periodicity of 14 years. Attempts to explain these vast fluctuations in numbers on the basis of climatic changes have been unsuccessful. At one time it was believed that these were caused by sunspots, and the sunspots and lynx cycles do appear to correspond during the early part of the 19th century. However, the cycles are of slightly different lengths and by 1920 were completely out of phase, sunspot maxima corresponding to lynx minima. Attempts to correlate these cycles with other periodic weather changes and with cycles of disease organisms have been unsuccessful. The snowshoe hares die off cyclically even in the absence of predators and in the absence of known disease organisms. The animals apparently die of shock, characterized by low blood sugar, hypoglycemia, exhaustion, convulsions and death symptoms which resemble the alarm responses induced in laboratory animals subjected to physiological stress. Visualizing this similarity, J.J. Christian, 1950, proposed that their death, like the alarm response, is the result of some upset in the adrenal pituitary system. As population density increases, there is increasing physiological stress on individual hares owing to crowding and competition for food. Some individuals are forced into poorer habitats where food is less abundant and predators more abundant. The physiological stresses stimulate the adrenal medulla to secrete epinephrine or adrenaline, which stimulates pituitary via the hypothalamus to secrete more ACTH, adrenocorticotropic hormone. This in turn stimulates the adrenal cortex to produce corticosteroids, an excess or imbalance of which produces the alarm response or physiological shock. In the latter part of the winter of a year of peak abundance, with the stress of cold weather, lack of food and the onset of the new reproductive season putting additional demands on the pituitary to secrete gonadotropins, the adrenal pituitary system fails, becomes unable to maintain its normal control on carbohydrate metabolism, and low blood sugar, hypoglycemia, convulsion and death follows. According to Kende 1974, several intrinsic factors such as disease, predation, food factor and natural selection affect animal cycles in addition to several extrinsic factors such as weather, solar radiation, and so on. POPUIATION Ecology and Evolution. Population parameters have been used by population ecologists to draw conclusions about evolution of species. R selection and K selection. In this regard, an important synthesis was done by MacArthur and Wilson, 1967, who declared that populations are outcome or K selection. The R-selected populations have a high intrinsic rate of growth R and tend to boom when environmental conditions are favorable and best when these conditions deteriorate. In consequence, they show great fluctuations in density and incidentally have the potential for large genetic change through the founder effect. The K-selected populations have relatively constant density at or near the carrying capacity K of the environment. RUAYRYOOH. Living creatures of an ecosystem form a biotic community. Biotic communities. Community ecology, communities, niche, and bioindicators. 1. The living entities of an ecosystem form a single biotic component, the community or biotic community. All the organisms of a community live together, share same habitat, influence each other's life directly or indirectly and have reached a survival level within a given radiant energy. Thus, a community is any assemblage of populations of living organisms in a prescribed area of habitat. A community is claimed to have one or more of the following attributes: 1. Co-occurrence of species, 2. Recurrence of groups of the same species, and 3. Homeostasis or self-regulation. Krebs, 1985. Community is a larger unit than the population and it achieves many characteristics that are not found in its constituents, i.e., the organisms in the populations. Communities may have a wide range of sizes, ranging from a small patch of land or water body to extensive forests. Minor communities are greatly influenced by inputs from adjacent communities, while major communities are relatively independent and self-sufficient of their habitat. Communities differ from place to place and at the same place at different times. The approach of botanists and zoologists to community studies is quite different. While the zoologist is mainly concerned with the functional relationships within a community, involving both the plants and animals, the botanists are generally concerned with community structure and the changes that undergo in time and space. Characteristics of a community. Like a population, a community has a series of characteristics such as follows. 1. Species diversity. Various species of plants and animals live in a community and exhibit species richness or species diversity. The study of species diversity is an essential component of community study. For animal communities, a study of age structure and growth pattern is important. While for plant communities, floristics, study of taxonomy, life forms such as herbs, shrubs, climbers, trees are important. Since seasonal changes occur in the appearance of plant structure and growth, periodicities and phenology equals seasonal succession in natural communities are significant parameters. 2. Growth form and structure. The type of the community is described by major categories of growth forms, e.g., trees, shrubs, herbs, mosses, etc. These different growth forms determine the stratification, or vertical layering of the community. 3. Dominance. Among several species present in a community, a few exert a major controlling influence by virtue of their size, numbers, or activities. These are called as ecological dominance or dominant species. 4. Relative abundance. Different populations in a community exist in relative proportions and this idea is called as relative abundance. 5. Trophic structure. Who eats whom? The feeding relations of the species in the community will determine the flow of energy and materials from plants to herbivorous animals to carnivorous animals. CIASSIFICATION of the CO munis. Communities have been classified by different ecologists from different viewpoints. In terms of the general growth, composition, shape, etc., of vegetation, and organisms associated with them, communities may be classified as forests, deserts, grasslands, tundra and so on. Likewise, according to the amount of water in the habitat, communities may be divided as hydrophytic in predominantly aquatic habitats, mesophytic in moderately moist soils and xerophytic in arid or dry conditions. Communities growing in condition of abundant light are called heliophytic and those growing in shade are called geophytic. Clements 1916 recognized the fact that plant communities are not always the same at any place and he classified the communities on two parallel lines, one in the process of change which are called seral communities and the others are called stable or climax communities. Further, the global community is an enormous mass of life, comprising all the plants and animals in the world. The global community is further divided into continental communities and oceanic communities, since due to great variability in climatic factors, an exhaustive study in such vast areas is practically impossible. Therefore, communities are often studied as biotic province. A biotic province can be defined as a considerable geographic area, over which the climate is relatively uniform though often modified by physiographic features. Dice, 1952. Since biotic province is an abstract community, so based on ecological criteria, the associations are studied as concrete communities. A concrete community can be defined as a specific area which can be observed directly and which is an assemblage of plants and animals that actually exists and from which some ecological data can be collected. Lastly, a stand is the largest concrete community, for example, a particular forest, river, swamp, meadow or lake that can be seen, observed, measured and worked over by the ecologist. A microstand. Isosmall localized area within stand. Thus, each individual plant or animal with its associated parasites, epiphytes and commensals are good examples of the microstands. Composition of community. Composition of community comprises the following parameters. 1. Size. Communities may be large or small. Larger one extends over areas of several thousands of square kilometers, as forests, others such as deserts, etc., are comparatively smaller with dimensions in hundreds of kilometers, and still others such as meadows, rivers, in hundreds of kilometers, and still others such as meadows, rivers, ponds, rocky plateaus, etc., covering a more restricted area. Very small-sized communities are the groups of microorganisms in such microhabitats as leaf surface, fallen log, litter, soil, etc. 2. Number of species. The number of species and population abundance in communities vary greatly. Charles Elton, 1927, calculated that the communities of certain British rivers included 131 species of invertebrates, in addition to fish, amphibians, populations of algae, protozoa, bacteria, and rooted aquatic plants. In contrast to this, he found a community on a sandy beach consisting of only 5 species of invertebrates. A meadow equals a rich pasture ground, especially beside a stream. On clay near Oxford, England included 93 species of invertebrates in the soil and on above ground vegetation. In India too, various ecologists recently have estimated the number of species in the composition of various Indian communities. M.C. Dash and his co-workers 1980, 1981 and 1984 studied the composition of a biotic community in some tropical pastures of Sambalpur, India, and found that it consisted of 22 grass and other herbaceous species, the primary producers, 5 species of earthworms, 25 of test-bearing protozoa, 15 of nematodes and 7 of microarthropods. The team of 1980, 1984 reported 9 species of grasses and other herbaceous species, 1 of earthworms, 11 of testate protozoa and 16 of nematodes in a tropical hill ecosystem of Sambalpur. At one side of River Ib, Orissa, the community consisted of 1 species of rooted plant, 32 of phytoplanktons, 20 of zooplanktons and 7 of fish. Carr et al. 1987, et al. 1988. 3. Dominance. In each community there occurs diverse species. All these species are not equally important, but there are only a few overtopping species which by their bulk and growth modify the habitat and control the growth of other species of the community, thus, forming a kind of characteristic nucleus in the community. These species are called the dominants. Generally in most of the communities, only a single species is particularly conspicuous and dominant, and in such case the community is called by the name of dominant species, e.g., spruce forest community. In other cases, there may be more than one dominance in a community, e.g., oak hickory forest community. According to Clements and Shelford 1939, dominance is most commonly expressed in the reactions of an organism on its habitat. Community dominants sustain the full impact of the climate or the environment but modify this effect for other organisms within the community by tempering light, moisture, space, and other conditions. Only those other organisms that find these modified physical conditions tolerable can exist within the community. Furthermore, dominants are ordinarily the most prominent species in the community, make up its greatest mass of living material, biomass, and serve as the major source of food, substrate, and shelter for the animals that are present. In a forest community, trees are dominant. They decrease light intensity, increase the relative humidity, intercept precipitation, monopolize most of the moisture and nutrients in the soil, decrease wind velocity and furnish shelter and food for animals. Grasses play a similar, though less conspicuous, role in prairie communities, sedges, rush and cattails in marsh communities, sagebrush in the arid habitat of the Great Basin, mussels and barnacles on a rocky seashore, and so forth. 5. Ecological amplitude. The range of environmental conditions which a taxon can tolerate is called ecological amplitude. The composition of a biotic community in any habitat is dependent upon the frequency of environmental conditions in that habitat and the ecological amplitude of species populations. Thus, the climate and other abiotic and biotic conditions of a habitat determine the type of community which survives and develops. Structure or stratification of company unity. The communities exhibit a structure or recognizable pattern in the spatial arrangement of members of the communities. Stratification forms one of the outstanding general principles of community structure. Organisms are distributed unevenly throughout the biotic community, resulting in stratification. The stratification in a community may be resulted by the following factors: 1. Specific tolerations and adaptations. 2. Chemical reactions taking place between the byproducts and physical stratification. 3. Other organisms take up temporary or permanent residence as a direct L.U_E. 6. 250. Mountains of Western North America. Response to the presence of initial residents. The stratification in community may be horizontal or vertical and is different in aquatic and terrestrial habitats. 1. Horizontal stratification. A community may be divided horizontally into subcommunities which are units of homogeneous life forms and ecological relations. This horizontal division constitutes the zonation in the LOL. L. 2. Lower on the shore, where the tides always flood, are living beneath the stone some annelids and nemertine worms, e.g., Linnaeus. Adjacent to low water mark are certain species of nares, shore crabs, etc. 3. Holes and cracks in the rocks are occupied by various annelid worms, crustaceans, small sea cucumbers, and rock-boring bivalves. 4. There are rock pools that enclose those animals which escape direct current of water. Sandy shore-dwelling marine animals are in great part burrowers such as sphenoid Prionospio, Aricia annelids Glycera, Nerina archiannelids Protodrillus, and crustacean Emerita, Albania. Populations which inhabit this substrata adjust to both sand and mud particles. This becomes evident in their respiratory adaptations and locomotory and feeding behavior. 2. Vertical stratification. Vertical gradients in environmental factors such as the availability of sunlight, the temperature, and so on bring about a recognizable stratification in water bodies, particularly in marine ecosystem Fig.7.3, such as deep water lakes. The oceanic region, i.e., the region of open sea beyond continental shelf, comprises two zones. 1. Bathyal zone, i.e., region of continental slope and rise, and 2. Abyssal region, i.e., area of ocean, deeps. In terms of light penetration, the ocean region is vertically also divided into an upper thin euphotic zone equals the light compensation zone, and a vastly thicker permanently darker zone, the aphotic zone, including bathyal and abyssal zones. The oceanic region also includes two more vertical zones, the benthic bottom and the pelagic whole body of water. The community of pelagic zone includes 1. Phytoplanktons, the producers such as diatoms, dinoflagellates, flagellates, green algae, brown algae such as sargassum, 2. Zooplanktons, the consumers such as foraminifera, radiolaria, annelid worms, swimming snails, jellyfishes, ctenophores, etc. 3. Nectins, these are free-swimming marine animals which are essentially independent of water movements. Nectins are consumers such as cephalopods, sepia, loligo, nautilus bony fishes, sharks, sea turtles, whales, seals, and seabirds. Minute organisms that float or swim on surface water are called neustins. Benthos are bottom dwellers of benthic environment. They are sessile forms such as limpets, chitons, mussels, oysters, sponges, corals, hydroids, anemones, bryozoans, crabs, lobsters, some echinoderms, and flat neritic fishes. High tide oceanic depth. Vertical street. Low tide oval tathoms. Roots, rhizomes, and organisms such as bacteria, fungi, protozoa, microarthropods, insect larvae, pupae, oligochaetes, and mollusks. In forest community, however, the vertical stratification is most complete. Chaetodon includes following 5 subdivisions. 1. Subterranean subdivision. 2. Forest floor. 3. Herbaceous vegetation. 4. Shrubs, and 5. Trees. Figure 7.4. In some tropical rainforests, 3 more subdivisions exist in the forest floor such as leaf litter layer, L layer, partially decomposed leaf litter layer, F layer, and a humus layer. Thus, based upon the light and relative humidity requirements, we find stratification in above ground parts. Ecological potential of a zone or strata determines the distribution of community. Fenton, 1947, has classified these communities as 1. Microbiota, which include bacteria, algae, fungi, protozoans, rotifers, nematodes, and mites. 2. Mesobiota, which include many nematodes, centipedes, millipedes, spiders, insects, etc. 3. Macrobiota, which include plant roots, earthworms, toads, lizards, snakes, rodents, and large mammals including man. He also classified these communities according to their feeding habits such as chemophagus, ectophagus, entophagus, predators, and shelterers. Characters used in community struc2a. Structure of plant communities can be determined by quantitative and qualitative features as follows. 5 trees. 4 A. Quantitative structure of plant communities. Quantitative characters of the community include such characters as frequency, density, abundance, cover and basal area, etc. 1. Frequency. Various species of the community are recorded by different phytosociological methods, by taking any sampling unit such as transect, bisect, quadrat, point center or point quarters, etc. Frequency is the number of sampling units, as percent, in which a particular species occurs. Thus, the frequency of each species is calculated in the following way. Number of sampling units in which a species occurred frequency percent equals x 100 total number of sampling units studied. After determining the percentage frequency of each species, various species are distributed among ronchires, 1934, 5 frequency classes depending upon their frequency values, table 7:1. Total number of individuals of the species in all the sampling units. Abundance equals number of sampling units in which the species occurred. 4. Cover and basal area. The above ground parts of plants, such as leaves, stems, and inflorescence, cover a certain area. If this area is demarcated by vertical projections, the area of the ground covered by the plant canopy is called canopy cover, foliage cover, or herbage cover. Canopy cover is a good measure of the herbage availability and is estimated by chart quadrat, line intercept, or point frame. Basal area refers to the ground actually penetrated by all stems and is readily seen when the leaves and stems are clipped at the ground surface. It is either measured 2.5 centimeters above ground or actually on the ground level. Basal area of a plant is measured by calipers, line interception, or point-centered quadrat method. It is one of the chief characteristics to determine dominance. Since plants differ in their growth form, so the relationship between herbage cover and basal area differs in different types of plants as shown in figure 7.5. For the measurement of crown cover, the diameter is taken at ground level, across the canopy perimeter passing by the main stem. The cover is expressed as a percentage. Based on their cover values, species are grouped into the following 6 groups. Group 1 less than 5%, Group 2 5-25%, Group 3 25-50%, Group 4 50-75%, Group 5 75-95%, and Group 6 95-100%. B. Qualitative characteristics of plant communities. These involves parameters such as physiognomy, phenology, stratification, abundance, sociability or gregariousness, vitality and vigor, life form, growth form, etc. All these parameters can be described and may be grouped in point scales. 1. Physiognomy. Physiognomy is the general appearance of vegetation as determined by the growth form of dominant species. Such a characteristic appearance is often expressed by single term. For example, observation of a plant community having trees and some shrubs as the dominance, clearly forced us to conclude that it is a forest. Similarly, on the basis of appearance of a community, it may be a grassland, desert, etc. 2. Phenology. The life history of a plant species involves seed germination, vegetative growth, flowering, fruit formation, seed maturation, leaf fall, seed dispersal and death. These events are different for different species and are recorded for the individual species. A. C. Study of the date and time of occurrence of these events is called phenology. In other words, phenology is the calendar figure 7.5. Diagram showing relationship between herbage of events in the life history of the plant. Cover and basal area in different types of growth. Environmental factors tend to influence forms, herbs, shrubs and trees of plants. The phenological behavior of a species. Population. These phenological events are customarily recorded diagrammatically, figure 7.6, monthwise and season-wise, and provide valuable information. These diagrams are called phenograms. It will be significant to apply phenology to those ecosystems where seasonal changes are not sharply defined, e.g., deserts. Phenology also includes the periodicity and aspect of the community. Periodicity of communities. As already described, all the major phenomena on Earth are cyclic. The communities being dynamic entities respond constantly to seasonal changes and exhibit a variety of periodicities such as seasonal rhythms, lunar and tidal rhythms, and many periodicities associated with the cycle of day and night, e.g., circadian rhythms. Ecologists tend to recognize 6 phases. Seasons in the annual cycle: prevernal, vernal, estival, estivation, autumnal and hymal periods. Such cyclic fluctuations can be observed in both abiotic and biotic factors of a particular environment such as follows. 1. Abiotic variability, i.e., cycle changes in temperature, moisture, wind speed and a number of other factors. 2. Biotic variability, i.e., flora and fauna influenced by the immediate abiotic environment. 3. Stratification. As described elsewhere, stratification of communities is the one way in which plants of different two phytophases and species are arranged in 5, 3, 1. It denotes the proximity of plants to one another. Plants generally grow as isolated individual, in patches, colonies or groups. Braun Blanket 1951 classified plants into the following 5 sociability groups. I. S. Plants. Plants, stems, shoots found quite separately from each other, growing singly. E. S. Plants. Plants growing in small groups, 4 to 5 plants. E. S. Plants. Plants growing in small scattered patches. I. V. S. Plants. Several bigger groups of many plants at one place. V. S. Plants. Pure populations of plants covering large areas. Interdependencies, another community feature which includes inter- and intraspecific competition, parasitism, symbiosis and commensalism, all of which have been discussed elsewhere in this book. New feature which includes inter- and intraspecific competition, parasitism, symbiosis and commensalism, all of which have been discussed elsewhere in this book. 6. Vitality. It is the capacity of normal growth and reproduction, which are important for successful survival of a species. In plants, the vitality is determined by the following parameters: stem height, root length, leaf area, leaf number, number and weight of flowers, fruits, seeds, etc. Plant species March, July, August. Sep. October. Dec. Heteropogon contortus. Solar glauca. Single quote. IR Betarandinella sp. A life form is the sum of the adaptation of the plant to the community. It is determined by the stature of the plant species, their spread and characters of life form. Ronkier classified plants into following 5 broad life form categories, figure 7.7. His classification is based on the position of parenting buds on plants and the degree of their protection during adverse conditions. I. Phanerophytes. The growing buds of these plants are naked or covered buds with scale, i.e., they are not O. E. Chamophytes. In perennial buds, organs or seeds from phanerophytes to therophytes. These plants, the buds are situated close to the ground surface and these buds get protection from fallen leaves and snow cover. Chamophytes commonly occur in high altitudes and latitudes, e.g., Trifolium repens which is found in North America. E. Hemicryptophytes. These are mostly found in cold temperate zone. Their buds are hidden under soil surface, protected in soil itself. In the warm season, the growth of aerial parts is marked. Their shoots generally die each year, e.g., most of the biennial and perennial herbs including grasses. IV. Cryptophytes or geophytes. In these plants, the buds are usually buried in the soil or in bulbs and rhizomes where food is stored to withstand long periods of adverse climatic conditions, i.e., freezing and drying. Cryptophytes include the hydrophytes, buds remaining underwater, helophytes, marsh plants with rhizomes under the soil, and geophytes, terrestrial plants with underground rhizomes or tubers. V. Therophytes. These are seasonal plants, completing their life cycle in a single favorable season, and remain dormant throughout the rest unfavorable period of year in the form of seeds. Therophytes are commonly found in dry, hot or cold environments, i.e., deserts. Life forms of animals. There have been several attempts to classify the life forms of animals, but no definite system has resulted. Remain, 1952, Krivoletsky, 1972. The major life forms more often agree with their taxonomy than do plants, but some life forms include representatives from several different taxonomic groups. These can be recognized encrusting forms, such as the freshwater bryozoa and plumatella, and some sponges. Coral forms, including grass, leaf, or shrub forms, radiate. Forms, such as colindrates and echinoderms, bivalve forms, snail forms, slug forms, worm forms. Crustacean forms, insect forms, fish, snake, bird, and four-footed forms. Each of these major types may be divided into narrower structural or behavioral types. For example, Osborne et al. 1903 subdivided four-footed mammalian forms into following types. 1. Aquatic, swimming, seal, whale, walrus. 2. Fossorial, burrowing, mole, shrew, pocket gopher. 3. Cursorial, running, deer, antelope, zebra. 4. Saltatorial, leaping, rabbit, kangaroo, jumping mouse. 5. Scansorial, climbing, squirrel, opossum, monkey. 6. Aerial, flying, bat. C. Synthetic characters. Synthetic characters are determined after computing the data on analytical characters, i.e., quantitative and qualitative characters, of the community. For comparing the vegetation of different areas, community comparison needs the calculation of their synthetic characters. Synthetic characters are determined in terms of the following parameters. 1. Presence and constants. The presence expresses the extent of occurrence of the individuals of a particular species in the community. The species on the basis of its percentage frequency may belong to any of the following 5 presence classes that were first proposed by Braun Blanket. I rare, present in 1 to 20% of the sampling units. 2. Seldom present, present in 21 to 40% of the sampling units. I1I, often present, present in 41-60% of the sampling units. IV, mostly present, present in 61-80% of the sampling units. V, constantly present, present in 81-100% of the sampling units. A species with high frequency is considered constant. The term constancy is used when equal sample areas are taken in each stand, and presence is used when the area of the sample varies from stand to stand. 2. Fidelity. Fidelity or faithfulness is the degree with which a species is restricted in distribution to one kind of community. Such species are sometimes known as indicators. The species have been grouped into the following fidelity classes, which were first formulated by Braun Blanket. I. Fidelity 1 or strangers. Rarely occurring species which either have arrived from other communities or are relics from earlier stages of succession. E. Fidelity 2 or indifference, or ubiquitous. Species which occur in any community without exhibiting any preference to any particular kind of community. E. Fidelity 3 or preferentials. Species which occur in several kinds of communities but are predominant in one. IV. Fidelity 4 or selectives. Species occurring frequently in one kind of community and sometimes also in others. Sometimes preferential and selective species are considered together as characteristic. V. Fidelity 5 or exclusive or true. Species which are completely restricted to one kind of community. 3. Dominance. Itis used as a synthetic as well as analytical character. Dobbenmeyer, 1959. The number of organisms sometimes may not give correct idea of the species. If one bases his conclusion on number, a single or few trees in a grassland, or few grasses in a forest should be of little value. But if he considers the species on the basis of area occupied or weight biomass, the situation may be different. Thus, in dominance, cover is included as an important character. The single tree in grassland may occupy fairly a large area and may have much mass. Relative dominance cover, RDO, is calculated in the following way. Dominance cover of the species. Relative dominance cover, RDO, equals versus COV, total dominance cover of all the species. 4. Importance value index, IVI, and polygraph construction. This index is used to determine the overall importance of each species in the community structure. In calculating this index, the percentage values of the relative frequency, relative density and relative dominance are summed up together, and this value is designated as IVI, or importance value index of the species. The IVI provides an idea of the sociological structure of a species in its totality in equals. The community but does not INRBS. __, 75 _. Decay. The term niche was for the first time used by Joseph Grinnell, 1971, to explain microhabitats of California thrashers. According to him, niche is the ultimate distributional unit, within which each species is held by its structural and instinctive limitations. No two species in the same general territory can occupy for long identically the same ecological niche. The area of a species or any other taxon refers to the total extent of its geographical range of dispersal. This can be plotted on a map. The habitat of a species described in a single word or phrase is the totality of abiotic factors to which the species is exposed in this area. Thus, one can talk of marine habitats, coastal habitats, marsh habitats, forest habitats, disturbed habitats, or even dry habitats. In many cases, however, the habitat of the species is highly specialized. Certain species of leaf miners, for example, live only in the upper photosynthetic layer of the leaves of certain species of plants, while other species live in the lower cell layer. These patterns of location are so consistent that it is clear that the habitats of the species are different. Subdivisions of the environment on this scale are commonly called microhabitats. Thus, the leaf constitutes a microhabitat for leaf miners within the total forest, and the different cell layers of leaf constitute different microhabitats within the leaf for different species of leaf miner. Conditions within the leaves are quite different from the general conditions in the forest. The specific environmental variables in the microhabitat of a population is called microenvironment or the microclimate. Clapham Jr., 1973. Though habitats and microhabitats indicate the places where organisms of a community live, but, if one wishes to understand how a species fit into the fabric of the ecosystem, it is not enough to merely describe the creature's habitat or microhabitat. He must also understand its functional role within the community, address, of the organism but its profession, as well. This includes what it eats, what it does, where it lives, everything about it that influences the community. Thus, the ecological niche is the property of the community and it represents the place of the species in the formal community structure. In fact, the ecological niche is an inclusive term that involves not only the physical space occupied by an organism, but also its functional role in the community, i.e., trophic position occupied, and its position in environmental gradients including other conditions of existence. These three aspects of ecological niche are generally designated as I, the spatial or habitat niche, the physical space occupied, E, the trophic niche functional role, i.e., trophic position, and E, the multidimensional or hyperbloom niche position in the environmental gradients. 1. Spatial or habitat niche. This type of niche pertains to the physical space occupied by an organism. O. Neal, 1967, has cited a good example of spatial niche in the distribution of 7 species of millipedes in forest floor of a maple oak forest. All the 7 species occur in the same general habitat, i.e., forest floor, and all belong to the same basic trophic level, i.e., all are decomposers in their role as detritivores. Each species of millipedes predominates in its own specific different microhabitat. In fact, there exist several gradients in decomposition stage from the center of log to the position underneath the leaf litter, and these gradients constitute distinct microhabitats in the same general habitat, the forest floor. Thus, Ureus erythropicus predominates in the heartwood at center of logs, Pseudopolydesmus serratus in superficial wood of logs, Narcissus americanus in the outer surface of the logs beneath the bark, Schistonotus granulatus under log but on the log surface, Fontaria virginiensis under the log but on ground surface, Cledogonia caesioannularis within leaves of litter, and Abatian lacterium beneath litter on ground surface. 2. Trophic niche. In this case, 2 species live in the same habitat but they occupy different trophic niches, because of differences in food habits. For example, 2 aquatic bugs, Notonecta and Coryxa, live in the same pond but occupy different trophic niches. The former is an active predator that swims about grasping and eating other animals, but the latter feeds mainly on decaying vegetation. Finches of Galapagos Islands in America provide another good example of trophic niche. These birds belong to 3 genera, viz., Geospiza, ground finch, Camarynchus, tree finch, and Certhedia, warbler finch. These all birds live in the same general habitat, but differ in terms of their trophic position. For example, in tree finches, one of the species, Camarynchus crassirostris, has a parrot-like beak and is basically vegetarian, feeding on buds and fruits. Rest of the tree finches are insect eaters, 3 of which feed on insects of different sizes. Another species, C. Heliobates, feeds on insects in mangrove swamps. The most interesting is woodpecker finch, C. pallidus, which climbs tree in search of insects in the cracks of bark. Although it has woodpecking beak, it lacks long extensive tongue of woodpecker. Notonecta. 3. Multifactor or hyperbloom niche. This concept, where niche is considered as an abstract n-dimensional inhabitated hyperbloom, was developed by G. E. Hutchinson, 1965, of Yale University. Suppose that we measure the range of some environmental factor, e.g., temperature, over which a particular species can live and reproduce, in effect, its range of tolerance, and we put this on a graph. Suppose that we then do the same for another environmental factor, e.g., humidity, and put this on the second axis of the graph. The space that is enclosed will represent the niche of the species. If the effects of 2 variables, or factors, i.e., temperature and humidity, are independent, the space would be 2-dimensional box, figure 7.9a. However, in fact, temperature and relative humidity are not independent in their biological effects. Thus, tolerance to higher temperature may be related with increase in relative humidity. Under such condition of interacting variables or factors, the niche instead of being the box-like would be an oval in shape, figure 7.9b. Now suppose that the tolerance to levels of a third factor, i.e., available phosphorus, was affected by interactions with both temperature and relative humidity. We would now have a niche representing 3 variables or 3-dimensional, volumetric figure, figure 7.9c. If there is present a 4th variable, the space enclosed would be a hypervolume with 4 dimensions. Since there are a large number n of other environmental factors, both abiotic and biotic, that affect the population, the niche is n-dimensional hyperbloom, an abstraction since one is able to draw only with respect to 3 dimensions. Thesis: the fundamental niche of the species. When the fundamental niches of 2 species overlap them, the 2 species are competing. Competitive exclusion would be called into play only in Coryxa. The complete overlapping of n-dimensional niches. Figure 7.9d. Thus, this con 2b. Sept refers to the totality of Euro range of community metabolism. Once a basic idea of community structure is gained, it becomes necessary to ask the question, how does a community function? The study of functional organization or community metabolism is fundamentally a study of food relationship within the community. Such studies involve investigations one flow of energy and cycling of nutrients within a community. Through the food web, one can make the graphic depiction of the actual flow of food materials from population to population through the community. Community stability. One of the most important considerations about communities is their stability, or lack of variation in time. There are two discrete senses in which this term is used. The first, community stability, refers to the degree of fluctuation in population size of the populations comprising the community. The second, environmental stability, refer to the fluctuations in the abiotic factors of the ecosystem. The two types of stability are closely related, and stable communities are generally to be found in stable environments, while unstable environments are more often characterized by unstable communities. The global range of variation in community stability is extraordinarily broad. Some communities, such as the tropical rainforest or the coral reef, appear exceedingly stable, and the densities of their populations are constant over long periods of time. Others, such as the tundra, are extremely variable, and they are made up of populations that fluctuate widely in abundance. Much of this fluctuation is due to variations in environmental factors which the community cannot avoid. However, genetically determined adaptations that have arisen through natural selection also play a part in determining the responses of a population to environmental fluctuations. Further, the structure of community in which a population is found has a major role in determining the degree of environmental variability to which the species is subjected. There are at least two mechanisms by which community structure affects the stability or variability of a community. The first mechanism involves the relationship between community structure and microenvironment. Many organisms are protected from the full impact of environmental fluctuations because of the presence of other members of the community. For example, the floor of a conifer forest is protected from winds, evaporation, and temperature shifts, making it a much less harsh and variable environment for organisms than the forest canopy or an open field adjacent to the forest. The second mechanism by which community structure affects community stability is community diversity. But about the correlation of community stability and community diversity, many ecologists have expressed their doubts. Hairston et al., 1968. Ecotone and edge effect. In the ecocline, i.e., gradient of ecosystems, the line of demarcation boundary line between two communities is often very difficult in view of the chances of overlapping of one community over another. Such demarcation will be conspicuous only when the dominance of the adjacent communities show clear and characteristic differences. The transition zone between two or more diverse communities is called the ecotone. The common examples of ecotone are following: the border between forest and grassland, the bank of a stream running through a meadow, or between a soft bottom and hard bottom marine community. An ecotone, thus, presents conditions that are intermediate to the communities which are on either side of it. The ecotones may be narrow or very wide, extending to large areas. The community on either side of the ecotone may have a typical structure, but the ecotone is strikingly different. The ecotone has a higher diversity than either of the main communities, a diversity that is not directly controlled by the climate or further fundamental environmental factors, but because of the migrations of individuals of different species populations from both communities. Further, a number of special populations can become adapted specifically to the ecotone, even when both of the major communities are too simple or otherwise unsuited for successful colonization by the species. This potential for the ecotone to act as habitat for species found in neither major community is termed as the edge effect. A common example of the edge effect in action can be seen in those species of owl that live in or near ecotones between forests and grassland. They depend on forest trees for nesting, and they do their hunting in the grassland, where they depend on field rodents for food. In man-made communities such as agricultural fields, the ecotone transition zone ecotone between the field and the forest may act as a refuge for animal species formerly found in the plowed area, as well as for other plants such as weeds. Ecotones of this type are also the prime habitat of many species of insect, game, bird, and mammal. Factor compensation and ECO types. Organisms are not just slaves to the physical environments. They adapt themselves and modify the physical environment so as to reduce the limiting effects of temperature, light, water, and other physical conditions of existence. Such factor compensation is particularly effective at the community level of organization, but also occurs within the species. Species with wide I species characteristic of community geographical ranges almost always develop locally adapted populations, called ecotypes, that have different IL species characteristic of the transition zone. Genetically based tolerance ranges and optima to correspond to the environments within which they normally live. Odom, 1971, Clapham Jr., 1973. The term ecotype was coined by a Swedish botanist, Goethe Teresin, in 1912, and in 1922 he defined it as the product arising as a result of genotypical response of an M species characteristic of community B. Among animals, ecotypes have been reported in jellyfish, Aurelia aurita bullock, 1955, European oyster, Austria edulus coringa, 1957, and frog. Some frogs, for example, undergo rapid development at lower temperatures at one extreme of their range of distribution, while others show a more retarded development rate at higher temperatures at another extreme of their range. ECOIOGICALINDICATORS. Since specific factors often determine rather precisely what kinds of organisms will be present in a particular habitat, we can turn the situation around and judge the kind of physical environment from the organism present. It is found that certain species of microorganisms, plants and animals, have one or more specific requirements, and they become very much limited in their distribution. Thus, the occurrence of such species in a particular area indicates special habitat conditions, and such species are called biological or ecological indicators, since they indicate some very specific condition of the environment. Some of the Indian plant species which serve as ecological indicators have been tabulated in Table 7-2. Table 7-2. Certain ecological indicators of plant species. Name of species indicator S of E R equals O underscore equals W N underscore equals equals Utricularia, Chara, Wolfia, Audelia elysiomoides. Petridium sp. Argemon maxicana. Rumix acetosella. Carissa spinarum and Capris spinosa enicosma literal. Saccharum spontaneum. Ziziphus rotundifolia. Andropogon scoparium. Dot. Lipia nodiflora and Rumix sp. Dot. For example, Escherichia coli, a bacterium, is an index organism for aquatic pollution. Some species of diatoms Arophidae indicate the degree of sewage pollution of lakes. The presence of fusulinids protozoans in the soil indicate the petroleum deposits. Similarly, certain animal species serve as ecological indicators. Chironomid larvae and maggots increase in number as the pollution increases. Burrowing mayfly hexagenia serves as indicator for well-oxygenated aquatic environments. Emigration and consequent disappearance of certain Indian fish species such as Catla, Labiogonius, Labiobata, Labio-rohita, Notopterus, etc., from Kalinadi River near Mansapur Sugar Factory, UP indicate industrial pollution of water, as these fishes are found to be very sensitive to aquatic pollution. Burma, 1971. RUAYNIO ENO 18 SNMNDK. The process of directional change in vegetation is termed as succession. Ecological succession. Hence, stripped of its original vegetation by fire, flood, or glaciation, an area of bare ground does not remain devoid of plants and animals. Beginning with plants, area is rapidly colonized by a variety of both plant and animal species that subsequently modify one or more environmental factors. This modification of the environment may in turn allow additional species to become established. The transitional series of communities which develop in a given area are called sera-orseral stages, while the final stable and mature community is called the climax. The development of the community TTY action of vegetation on the environment leading to the establishment of new species is termed succession. Succession is the universal process of directional change in vegetation during ecological time. It can be recognized by the progressive change in the species composition of the community. Retrogression in community development is not occur unless succession is disturbed or halted by fire, grazing, scraping, or erosion. Causes of succession. Since succession involves a series of complex processes, so there exist many causes of its occurrence. Ecologists have recognized the following three primary causes of succession. 1. Initial or initiating causes. These are climatic as well as biotic in nature. The climatic causes include factors such as erosion and deposits, wind, fire, etc., which are caused by lightning or volcanic activity. The biotic causes include various activities of organisms. All these causes produce the bare areas or destroy the existing populations in an area. 2. Esesis or continuing causes. These are processes as migration, esesis, aggregation, competition, reaction, etc., which cause successive waves of populations as a result of changes, chiefly in the edaphic soil features of the area. 3. Stabilizing causes. These include factors such as climate of the area which result in the stabilization of the community. Trends of succession, functional changes. Trends of changes during ecosystem development from a young to mature stages include the following features. 1. A continuous change occurs in the kinds of plants and animals. 2. An increase in the diversity of species takes place. The general appearance of the community or the physiognomy keeps on becoming more and more complex as the succession proceeds. 3. There is a progressive increase in the amount of living biomass and dead organic matter. Such an increase occurs in gross as well as net primary production in the initial and seral stages. Thus, there is more biomass accumulation, gradually reaching a huge biomass structure in the climax. 4. Green pigment chlorophyll go on increasing during the early phase of primary succession. 1. Trends of changes during ecosystem development from a young to mature stages include the following features. 1. A continuous change occurs in the kinds of plants and animals. 2. An increase in the diversity of species takes place. The general appearance of the community or the physiognomy keeps on becoming more and more complex as the succession proceeds. 3. There is a progressive increase in the amount of living biomass and dead organic matter. Such an increase occurs in gross as well as net primary production in the initial and seral stages. Thus, there is more biomass accumulation, gradually reaching a huge biomass structure in the climax. 4. Green pigment chlorophyll go on increasing during the early phase of primary succession. The ratio of yellow-green pigments remains around 2 in the early stages and increases to 3 to 5 in the climax stage. Pigment diversity also increases. 5. The community respiration increases but the PRIE, production-respiration, ratio remains more than 1 in the seral stages. The huge living biomass respires a lot in the climax stage and the PR ratio equals 1, i.e., PR equals 1. Thus, in the early stages P greater than R and in the climax stage P equals R. 6. The food chain relationships become more complex as succession proceeds. 7. Nutrients in the young stage are allocated mostly in the soil, but as the seral stages advance, nutrients get allocated more in the vegetation and less in soil. Further, the nutrient cycling becomes more closed or intrabiotic with an efficient cycling mechanism, whereas in the young stage the nutrients easily leak out from the system, i.e., the cycling is more of an open type. 8. The role of detritus becomes progressively more and more important. 9. The quality of the habitat gets progressively modified to a more mesic condition from either too dry or too wet condition, in the early seral stage. 10. The niche specialization increases, i.e., different functions are more effectively performed by specialist species in mature seral stage, whereas in early stage many functions are performed but less efficiently by a few species. 11. The life cycle of mature community species are longer and more complex. 12. The importance of macroenvironment becomes less in later stages. 13. Relationship among component species becomes more and more mutualistic or helpful even though there exist enough competition and allochemic activities to prevent invasion of outside elements. 14. Dispersal of seeds and propagules is by wind in young stage, while by animals in mature stage. Basic types of succession. Based on different criteria, there are following kinds of succession. 1. Primary succession. If an area in any of the basic environments, such as terrestrial, freshwater or marine, is colonized by organisms for the first time, the succession is called primary succession. Thus, primary succession begins on a sterile area, an area not occupied previously by a community, such as newly exposed rock or sand. Thus, primary succession begins on a sterile area, an area not occupied previously by a community, such as newly exposed rock or sand dune where the conditions of existence may not be favorable initially. 2. Secondary succession. If the area under colonization has been cleared by whatsoever agency, such as burning, grazing, clearing, felling of trees, sudden change in climatic factors, etc., of the previous plants, it is called secondary succession. Usually the rate of secondary succession is faster than that of primary succession. Succession because of better nutrient and other conditions in area previously under plant cover. 3. Autogenic succession. After the succession has begun, in most of the cases, it is the community itself which, as a result of its reactions with the environment, modifies its own environment and, thus, causing its own replacement by new communities. This course of succession is known as autogenic succession. 4. Allogenic succession. In some cases, replacement of one community by another is largely due to forces other than the effects of secondary succession. Communities on the environment. This is called allogenic succession and it may occur in a highly disturbed or eroded area or in ponds where nutrients and pollutants enter from outside and modify the environment and in turn the communities. 5. Autotrophic succession. It is characterized by early and continued dominance of autotrophic organisms such as green plants. It begins in a predominantly inorganic environments and the energy flow is maintained indefinitely. There is gradual increase in the organic matter content supported by energy flow. 6. Heterotrophic succession. It is characterized by early dominance of heterotrophic organisms such as bacteria, actinomycetes, fungi and animals. It begins in a medium which is rich in organic matter such as small areas of rivers, streams, these are polluted heavily with sewage or in small pools receiving leaf litter in large quantities. 7. Induced succession. Activities such as overgrazing, frequent scraping, shifting cultivation or industrial pollution may cause deterioration of an ecosystem. Agricultural practices are retrogression of a stable state to a young state by man's deliberate action. 8. Retrogressive succession. It means a return to simpler and less dense or even impoverished form of community from an advanced or climax community. In most cases, the causes are allogenic, i.e., forces from outside the ecosystem become severe and demanding. For example, most of our natural forest stands are degrading into shrubs, savanna or impoverished desert-like stands by the severity of grazing animals brought from surrounding villages. Excessive removal of wood, leaf and twig litter also leads to retrogressive succession. 9. Cyclic succession. It is of local occurrence within a large community. Here cyclic refers to repeated occurrence of certain stages of succession whenever there is an open condition created within a large community. Further, in ecological literature, there are mentioned still so many other kinds of succession, depending mainly upon the nature of the environment where the process of succession has begun. Thus, it may be a hydrous or a hydrarch when succession occurs in regions where water is in plenty, as ponds, lakes, streams, swamps, bogs, etc., a mesarch when there are present adequate moisture conditions, and a xerosar or xerarch when there is present minimum amount of moisture, e.g., deserts, rocks, etc. Sometimes, few more categories are recognized such as lithosar, succession initiating on rocks, samosar, succession initiating on sand and hollowsar, succession occurring in saline water or saline soil. GENERALPROCESS of succession. The entire process of primary autotrophic succession is completed through the following sequential steps. 1. Nudation. This is the development of a bare area without any form of life. Exposure of new surface may occur due to several causes such as landslide, erosion, deposition, or other catastrophic agency. These causes of nudation are of three main types. I. Topographic. Due to soil erosion by gravity, water or wind, the existing community may disappear. Other topographic causes include deposition of sand, landslide, volcanic activity and other factors. I. Climatic. Glaciers, dry period, hails and storm, frost, fire, etc. may also destroy the community. E. Biotic. Man forms a most important biotic factor. He is responsible for destruction of forests, grasslands for industry, agriculture, housing, etc. Other factors are disease epidemics due to fungi, viruses, etc., which destroy the whole population. 2. Invasion. Invasion is the successful establishment of a species in a bare area. The species actually reaches this new site from any other area. Invasion includes the following three steps. I. Migration, dispersal. The seeds, spores, or other propagules of a species reach the bare area. This process is called migration and is generally brought about by air, water, etc. E. Esosis, establishment. After reaching to new area, the process of successful establishment of a species, as a result of adjustment with the conditions prevailing there, is known as esosis. Esosis is followed by full-scale colonization. In plants, after migration, seeds or propagules germinate, seedlings grow, and adults start to reproduce. E. Aggregation. Colonization by successive offspring and new migrants help increase the population, a process called aggregation. Thus, after esosis, as a result of reproduction, the individuals of the species increase in number and they come close to each other. Plants are autotrophic organisms which are the first to colonize and aggregate are called pioneers. The pioneer communities are likely to be more dynamic and have low nutrient requirements and to take minerals in comparatively more complex forms. They are small-sized and make less demand from environment. 3. Competition and coaction. Due to aggregation of a large number of individuals of a species at the limited place, there develops competition, i.e., interspecific and intraspecific competition for space and nutrition. Individuals of a species affect each other's life in various ways and this is called coaction. The species which fail to compete with other species are ultimately discarded. The reproductive capacity, wide ecological amplitude, etc., help the species to withstand the competition. 4. Reaction. Reaction includes mechanism of the modification of the environment through the influence of living organisms on it. Due to this very significant stage, changes take place in soil, water, light conditions, temperature, etc., of the environment. As a result of reaction, the environment is modified and become unsuitable for the existing community which sooner or later is replaced by another community, seral community. The whole sequence of communities that replaces one anatherant given area is called a sera, and different communities constituting the sera are called seral communities. Seral stages are developmental stages. 5. Stabilization climax. Finally, there occurs a stage in the process when the final tear 6, O, O, and EW and O, O, O